So, hello everybody. My name is John Johnson from NSF Pharma Biotech. This is my webinar on an update to Udrillex Volume 4 Annex 1 and how to generate a contamination control strategy, obviously to help you identify and mitigate risks in your sterile production unit. Of course, many of you on the call today will know an awful lot about NSF Pharma Biotech. We are experts in specialist training associated with GMP, GXP subjects across the industry. We have a very well-established expertise in auditing, including certification, vendor assurance, and GMP regulatory inspection preparation. And of course, we're involved in transformational projects, including quality culture changes, GMP improvement projects, new facilities, new builds, and business improvement programs. As I mentioned, I'm John Johnson. I'm a Vice President here at NSF Health Sciences. My experience across the industry over the last 35 years has been associated largely with both quality and manufacturing units, sterile and non-sterile facilities, CMO and big pharma across the globe in a number of operational and corporate leadership roles. So, the content of this webinar, well, why is Annex 1 being updated? What are we hoping to achieve when the updates finally go through their final approval? And also specifically pulling out one of the new expectations from Annex 1, how do you perform a contamination control strategy document? How do you generate one? How do you use it? What are the common pitfalls in generating such a document? And how would you use it to actually improve the product quality and GMP compliance associated with your steriles production unit? Well, here's a quick reminder on why it was updated in the first place. Just taking into account the science, the compliance, the leadership, and the education associated with sterile production. First of all, the science. So this will be the first major update since its inception in 1996, and of course there's been a number of new technologies, new approaches, new components and equipment put in place in that time period. So this seeks to reflect those new technologies in common use in the industry right now. There was also a compliance-related concern that the Annex wanted to deal with. This was around a perception of risk that had grown across the industry, partly due to the proliferation of global supply chains with maybe some fairly tenuous global oversight processes in place across some of these complex supply chains. There's also a need for the regulatory bodies to clarify some of these expectations, to try and minimize the variation in how Annex 1 is interpreted and implemented in practice. There was also a concern that there were some notable repeat GMP deficiencies across the industry that had not been identified, assessed, and mitigated successfully. So there was a need to try and do something in Annex 1 that would make it clear what was acceptable and what was not acceptable. And ultimately, and a key message for this webinar, is Annex 1 needed to align better with ICH-Q9 quality risk management and also ICH-Q10 pharmaceutical quality systems. There was also a concern across the industry that there had been a lot of turnover of expertise in the last 10, 15 years. And this had led to some real stresses and strains within the industry where the level of subject matter expertise within this sector of the pharmaceutical industry had not been maintained to the same level. So there was a need to clarify some of these particular requirements. Also, the regulators had noted that a number of internal education programs that they had reviewed on site had not been derived and aligned with the risks to the product quality. So there was a lack of detail in the education program, a lack of practical expertise, a lack of competency or proficiency testing, and ultimately some of the education programs had failed to really explain the know why as much as the know how. So what did the updates aim to achieve? Well, they wanted to reflect new technologies, they wanted to clarify some expectations, remove some ambiguity, and to add some scientific rationale to the design, control and monitoring of sterile processes, and try to encourage and catalyze better thought as to how science and risk management can be used to properly define your design, your control and your monitoring processes. Ultimately, of course, to improve the quality of sterile products and the industry's ability to remain within GMP. So you may remember this slide from previous webinars or previous NSF courses. This gives the 12 sections of Annex 1 and lists out some of the major changes that are coming in the new draft. And I just wanted to draw your attention to some of the key changes here. So the first one, top right there, is all about trying to emphasize the importance of staff behaviors during aseptic technique and during sterile production, and to really ensure that the education of your staff is not just around training, transactional, one-off courses, but is associated with good design, good behaviors, user-centered input, and proper engagement with your staff. Because of all disciplines, of all dosage forms, sterile production still depends on what's between the ears of our operators, engineers, QC staff, production and QA team. Moving on to section 7, it also really wants to emphasize the need to separate the key source of contamination, i.e. ourselves as operators, to separate operators from the process using restricted access barriers and isolators, and to emphasize the importance that any ABA transfers and any instances of operators standing within laminar flow, grade A environments is going to be prohibited under the new Annex 1, to make very clear that separation of staff and operators from open products and critical zones within the sterile production unit is absolutely critical. It also sought to make some clarification on the requirements for pre- and post-use filter integrity testing, so-called PUPSIT, pre-use post-sterilization integrity testing. And the indications are that the new Annex 1 will still require this as a EU CGMP expectation, that there will be a mandatory need for pre- and post-use integrity testing. There's still some discussion about this, there's still some input from industry required, but this is the current thinking from the agencies in Europe. The two areas I've coloured with red stars there really are the segue into the main part of this webinar, and what the Annex is looking to do here is to use risk assessment techniques in a much more proactive way to actually help you not just with the reactive issues when issues occur, but also to be able to predict, preempt, assess the risk and mitigate the risk before they become a crisis. Not least to use risk assessment techniques to develop your environmental monitoring program, but actually more importantly, to make an assessment of your facilities, utilities, equipment, people and processes so that you can see where risk occurs and do whatever is necessary to mitigate those priority risks. So the key messages shown on slide 8 here. Key link to ICHQ 9, an absolute requirement to use effective root cause analysis and scientifically justified proactive product impact assessment, to really focus on your education programs of your staff, all levels and all disciplines, creating the right behaviours and engagement of the staff and attitudes of the staff, separating the staff from open or critical stages, and also some consideration of new technologies or non-mainstream processes. So we've reviewed the draft Annex 1 with our NSF team of experts, and we felt that there was going to be a need to improve the Annex 1 draft to the next level, to really take into account more detail on particularly the following 4 items. So the first item critically is to have a much clearer set of guidance on whether there is a mandatory need to comply with EN 285. So EN 285 is the European norm that deals with the quality of clean steam, the design, control and monitoring of clean steam systems, specifically associated with those that supply steam sterilizers. And also, of course, an understanding of how to operate, monitor and provide performance checks on your steam sterilizers. And we felt that there ought to be more detail in what to comply with, how to comply with it, and a reference to EN 285. Secondly, we wanted to see more clarification about the use of VHP as a surface sanitization method, and clarification that regulators do not agree that VHP is a suitable sterilization method for product contact surfaces. So the risk with VHP is that, of course, it's not penetrating, and it also depends upon meticulously clean surfaces, and is not considered a suitable sterilization method by some regulators. So we wanted clarification on how VHP would be viewed by the regulators. We also felt that there should be some more information about the validation of disinfectants, specifically how to validate them, how to perform ongoing monitoring of them, what types of flora, standard flora and also cleanroom flora would be chosen, and what kinds of specifications for the validation would be expected. We also felt with the prevalence now of single-use disposable systems, there ought to be some guidance on the design, use and monitoring of single-use disposable systems within sterile production units. There was also some detail and nomenclature that could be clarified, and ultimately NSF are going to suggest those clarifications to the agency by the deadline of 20th of March. So, of course, in order to have a proactive approach to quality risk management, you need some evidence of actually taking the right decisions with the right information at the right time with the right intentions. And that's relatively easy to assess by assessing technical reports, GMP documents, deviation reports, periodic quality reviews. But what actually drives those key decisions? What is it that makes the difference? We see the difference being staff behaviours. And staff behaviours are also then underpinned by their skills, their ability, their competency, their beliefs, what they value, and how they identify themselves within the production process and the supply chain process. But actually, all of these things will not be successful unless you have alongside a proactive strategy for risk management. So, how can you better manage risk in a sterile production unit? How can you do it proactively? So a key Annex 1 requirement is now to generate a contamination control strategy that ensures all risks are identified, all risks have a standard way of assessment, and once they're assessed, the priority risks are then properly mitigated. So, if I'm going to generate this document, how do I make sure this document is credible and useful? Well, we are really passionate about using a multidisciplinary team to generate that document. We feel that everybody from engineers, production, QA, microbiology, QC, technical department should be involved in identifying and mitigating risk from your production unit. We know that we get a much better perspective when multiple perspectives are employed. We also realize that in order to really get a report that is going to be useful and actionable, you need to have the authority, freedom, and insight to actually speak out and project the brutal reality of your unit. So if the unit is not well designed, is old-fashioned, obsolete, is creaking, is aging, is not performing at the level expected, has had difficulty during validation, or has had recurring deviations during environmental monitoring, all of those things need to be called out in the contamination control strategy, and the staff involved need to feel the freedom to be actually speak out and call out the brutal reality of how that unit is really performing. And that requires you to have a blame-free culture and a very engaging and inclusive method of generating this insight. We see process flows and mapping processes to be very, very useful in assessing and identifying risks. We, of course, want you to use the standard scoring system in ICHQ9, where you help score each of the risks against the likelihood of high severity, high reoccurrence, and high detectability. And I'm going to explain that a little bit better in a moment. Then, of course, against a number of key attributes of your unit, you're then going to assess your design, control, monitoring, and precautions against risks of contamination, whether it's microbial or non-viable or chemical, risks of misbranding, and also risks to the compliance to UDRELEX Annex 1. And ultimately, once the scoring of each of those risks is codified, you can then look at the scoring system and, as a first priority, look to redesign that process, that facility, that utility in a way that improves the level of risk management as a 100-year fix, not as a sticking plaster, but something that will endure and be robust for the long term. So if you go to slide 13, there's now 3 tables that try to describe from the draft Annex 1 how you might structure a contamination control strategy. So if you look at column 1, you'll see the various sections across the 3 slides from A to O that gives you the subject areas that are required to be assessed. We've shown then in column 2 who we would assign as a single point accountability or an owner of that section of the CCS, obviously leading the multidisciplinary team that we spoke about before. We've then in column 3 identified some of the key tools, key source documents, and key techniques for identifying risk. Remember, you may have the best risk management process in the world, but if you've not looked hard enough for risk, if you've not looked for the signals, if you've not got an open, blame-free culture that allows people to surface the concerns, then of course you're never really going to be able to mitigate risk to a suitable level. So it's important to be rigorous in the sourcing and identification of the risks. And for each of the sections, I've suggested some source materials, source techniques, and documentation that you could review to get that insight. Then in column 4, I describe essentially how then to codify and describe the key risks, the current precautions, the current tests and checks that you perform, and also identify from that free-form description some of the key concerns. Once they're all individually assigned, you can then, utilizing your multidisciplinary team, look at assessing the score of each risk. So remembering to score between 1 and 10, but to avoid the score of 5 so that nobody can sit on the fence and hedge their bets and just score everything as a medium figure. And then by multiplying the severity score, the occurrence score, and the detectability score, just as it's shown in ICHQ-9, it will then give you a priority list that you can then assign a team to generate smart kappa, which is specific, measurable, achievable, realistic, and target-orientated, making sure that any kappa that has multiple steps or multiple resources or certain risks associated with it has a project charter assigned to it so that you can see the glide path to the improvement that you seek. Part 2 shows the next set of sections, and obviously following the same process of owner, risk methods, risk description, risk score, and kappa. You can then codify for each of the sections. Moving on to slide 15, part 3, you can then utilize exactly the same methodology across those key sections. So this all sounds very logical, and of course it is very logical, and it's very proactive, and it will assess risk and surface risk for you, and obviously help you prioritize risk and then drive improvement. But why didn't we do this years ago? Why? Well, the best companies did and have done for a while, but there's many companies that have really only done this piecemeal and not taken a holistic or scientific deep dive approach to this. Why? Well, some companies really fear having a risk register. They fear what it's going to find, or they badly understand the return on investment and the benefits for actually surfacing, codifying, and better understanding risk. After all, this is an absolutely key part of knowledge management, and the education programs that your staff are involved in really benefit from everybody understanding the risks and precautions associated with every key step. There's also a real concern that many of the risk management tools that are in ICHQ-9 have been used really to either defend or justify the indefensible or the unjustifiable. So where there's been bad design or poor practice or unpredicted issues, sometimes those tools have purely been used to justify the status quo with no further action taken. And that clearly is a very bad missed opportunity, Ultimately, if you're going to identify risk, the worst thing that can happen next, of course, is you take no action, because you're essentially saying, I'll live with it. And there may be some risks that you simply can't live with because the size of regret, the consequences of the risk coming to pass are so significant to your business continuity, to your GMP compliance, or the product quality, patient safety aspects of what you do. Fundamentally as well, many firms have missed the opportunity to think in a joined-up way, to actually think about how this type of approach can improve business continuity, how it can reduce waste, and obviously ultimately improve CGMP. So, what are some of the common problems that we're seeing in the industry just now in this area? Well, ultimately, even when firms have put together a detailed risk management approach, sometimes we're finding that the staff involved have a lack of authority to take action, that they're unable to effect the change that they desire or so badly need. And of course, if you're going to embark on this type of program, you need people that are empowered and have the resources to take the necessary actions. There's also, we've seen in some quarters, a lack of appetite to change, that sometimes purely retraining, purely more checks, or purely more QC tests is never going to actually prevent a recurrence of that problem occurring. The problem needs preventing at source by a proper redesign or a replacement of facilities, utilities, or equipment. And we haven't always seen appetite to make those types of investment or make those types of changes. So therefore, people live with an unacceptable level of risk. We've also reviewed a number of quality risk registers and contamination control strategies where the companies have just made the whole thing way too complicated and have not been able to really see the wood for the trees, that they've really hidden and buried some of the key risks in a plethora of low-level concerns. And that obviously leads to a degree of stasis. Of course, what the organization needs when embarking on this kind of program is the individual, departmental, and organizational courage to call out the concerns, to actually codify and share those concerns, and then hold ourselves as individuals or departments or organizations to account, and then seek effective risk mitigation. So you really do need a culture and set of behaviors that will call out those risks and then take the necessary action. Of course, that necessary action has got to be something that leaves you with a perpetual level of GMP compliance. It needs to lead to an enhanced level of performance in your unit. And in order to achieve that, sometimes the actions are not necessarily straightforward or short in duration. So you need a certain degree of long-term thinking, strategic thinking, that allows you to make iterative improvements to reduce risk over the long term. And basically, short-term thinking is just not going to cut it. So what about if I don't actually write one of these, if I don't actually engage in this? Well, obviously, the first thing is you're going to miss the opportunity to reduce the risk of major concerns in your production unit. You're also going to miss the opportunity to make war on waste. You're going to miss the opportunity to reduce the 7 forms of waste in your unit and always suffer, unpredicted and at the worst possible time, the types of issues that make us go grey and make us toss and turn and not sleep at night. But ultimately as well, of course, once Annex 1 is approved, failing to have a contamination control strategy is actually going to be seen as a GMP violation. So, some next steps. Well, we talk a lot about the 6-fingered hand, about how these areas are so crucially important to the health and well-being of your pharmaceutical quality system. And all 6 of those are areas where you need expertise to PhD level. So let's be proactive. Let's not let the hand sleep. Let's get to work and start proactively identifying risks and doing the right thing for the long-term future. We also think, of course, a key next step is for you to get your comments in on Annex 1 to the agency by the 20th of March deadline. That's always going to be helpful. They ask for feedback and we should obviously provide it. I would also say that performing a line-by-line gap analysis of your operation against contamination risks, against the sources of contamination, or indeed other types of GMP or operational risk is crucially important, allowing you to have a risk register, a business continuity plan, a quality strategy, and a quality improvement plan. Of course, if you need more detail on how to do that, or some horsepower that will help guide and support your process in doing that, we have experts within NSF who can help you with both auditing, education, and consultancy support in that area. There's an awful lot of things we can help you with, shown on slide 20, but fundamentally, what we'd like to help you with is that change curve. So whenever there's a new regulation or a directive or a new expectation, a new part of UDRELEX or a new annex, the first stage when it comes out is denial. People, particularly if they've not been part of the generation process or review process or have been unaware of the change, denial and anger and rejection is the first step. The sooner you can get to realization and acknowledgement, move through the resistance stage and get into the planning and execution so that you can internalize the types of improvements that you need. The sooner you can get to that, the better. And sometimes you just need third-party support in that change curve to get you into a place where you can comply with the expectations. I'll draw your attention finally to 4 events that we've got coming up this year that are going to go into a lot more detail on risk management and contamination control strategies. So that's 2 events of A to Z of Steriles, which is in Manchester in April and Amsterdam in November. Quality risk management for Steriles in York in June, and pharmaceutical microbiology in April in York. Also draw your attention to our free 2018 webinar program. We're about 20% of the way through the program, and we've got some fantastic speakers and hopefully some reliable technology to be able to get that to you with clarity and value. So thank you very much for taking an interest in this key subject. I hope this has been useful. As you can see on the final slide, here's my contacts. Please feel free to drop me a line or an email, and I'd be very happy to give any insight or references and listen to your concerns. Please be aware of our free resources that are available on the website. We have a range of great webinars and videos there. They're all completely free of charge, and you can either go to our website or obviously access those through our NSF